Hi everyone. In this lecture, I'm going to talk about the glycolysis pathway, and it is also to be called as Embden Meyerhof Parnas pathway. So we know that whenever we ingest a food that travel all along our GI tract and the various enzymes work upon it to degrade it in a smaller molecules. So whenever we ingest carbohydrate and the metabolism is going on, then it breaks into a smaller component, that is monosaccharide. In form of glucose, it get into the cell and the oxidation process occur through various enzymes and it releases some sort of energy in form of ATP, and there our cell utilizes these energy. So here the glycolysis pathway is the 10-step reaction that occurs in all the living cells and it can be occur in aerobic condition or in anaerobic condition. It means in presence of oxygen aerobic and in absence of oxygen is anaerobic. But the end products are different. When it works aerobically, it forms 2 molecules of pyruvate, but when it works in anaerobic condition, that forms 2 molecules of lactate. So this glycolysis pathway provides energy to mature RBCs as well, and the intermediate, the substrate and the metabolites in this glycolysis pathway helps in synthesis of many non-essential amino acids, glycerol and fatty acid as well. So this is very important pathway for our body as well because it provides energy to our cell in form of ATP. So here we'll discuss how this glycolysis pathway runs, what all enzymes work upon it, and how this releases the energy in form of ATP, and what all hormones are responsible for its inhibition and activation. So that regulation we'll also see here. So first we'll see that this glycolysis pathway is run in 3 phases. That is, first is energy consuming phase, utilizing phase, second is splitting phase, and third is energy producing phase. So energy consuming, before this glycolysis process run and the glucose get oxidized, it start using some energy in form of ATP. So before it release ATP, it start consuming ATP to get further oxidized. So let's see what all substrates, intermediates and enzymes are there. So the first reaction in the first phase of energy consuming utilizing phase is the glucose get converted into glucose 6-phosphate. So glucose get catalyzed into glucose 6-phosphate and the catalyzing enzyme is hexokinase and or it may be glucokinase. So the enzyme hexokinase is available in all the living cells. In every cell in the cytosol, the hexokinase is there. But if this reaction is occurring in liver, then this enzyme is not there. So there the other enzyme work and catalyze this reaction is the glucokinase. So glucokinase primarily present in the liver that help in glycolysis in the liver. So the glucose, the 6-carbon molecule monosaccharide convert into glucose 6-phosphate. So here you can see the phosphate group is adding on carbon number 6 and the enzyme that catalyze this reaction is hexokinase. So the important thing is here is the kinase enzyme is always help in phosphorylation, that is adding of phosphate group. So from where the phosphate is coming, here in this reaction this glucose molecule consumes ATP Here it is utilizing the energy that is in form of adenosine triphosphate, and from this 3 molecule of phosphate, 1 phosphate is donating to this glucose, and there on 6 carbon the phosphate group get attached and forms glucose 6 phosphate. So ATP is converting into ADP. So this is the first reaction, and the very important thing here is this, that here this reaction is irreversible. It means here these enzymes are not helpful to converting this glucose 6 phosphate back into the glucose. So this reaction here is the irreversible. Now the next second reaction is the glucose 6 phosphate is converting into fructose 6 phosphate. So both glucose and fructose are hexoses. It means they both have 6 carbon in their structure. Now again the one more similarity is there, they both are isomer to each other. They have same chemical formula, but they differ in their arrangement of the atoms. So they are isomer to each other. So the enzyme that catalyzes this reaction from glucose 6 phosphate to fructose 6 phosphate is phosphohexose. Phosphohexose means both have 6 carbon. Phosphohexose isomerase. So this enzyme catalyzes this reaction and convert into isomer form fructose 6 phosphate. Now in next third reaction, the fructose 6 phosphate is converted into fructose 1,6 bisphosphate. So you can see here almost it is same, but here the 1 is also added, and before this phosphate I have written the bis. It means here the 1 molecule of phosphate is added on carbon number 1. So thereby it is called fructose 1,6 bisphosphate. So here the phosphate groups are 2, but 1 is on 6, 1 is on 1. So there are gap in between these 2 carbon. So thereby it is called bis. And this reaction is catalyzed by the enzyme phosphofructokinase. So here this kinase enzyme again helps in phosphorylation, and phosphorylation occurs from high energy yielding molecule ATP. From this triphosphate, it donates its phosphate group to this intermediate. Thereby the 1 phosphate group is attached on carbon 1 of fructose, and it is converting into ADP. ATP is converting to ADP, that is tri to diphosphate, and 1 phosphate is on carbon 1. So thereby it is called fructose 1,6 bisphosphate. Now again the important thing is here is the third reaction is also irreversible in nature. It means it cannot be converted back into this intermediate with the presence of this enzyme. So thereby it is irreversible reaction. So till now you can say that this is the phase 1. It means the energy consuming or utilizing phase, where the glucose get oxidized by using 2 molecule of ATP. So from glucose to fructose 1,6 bisphosphate, the molecule is utilizing 2 molecule of ATP to get further oxidized. Now the second phase comes, that is the splitting phase. Splitting, it means here the structure contains 6 carbon, and now this structure split into 2 molecules, and that both molecules contain 3 carbon in each. So one is forming is dihydroxy acetone phosphate, and the other molecule is glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. So they both have 3 carbon in each. So thus this 6 carbon component is split into half, 3 in each, and both have phosphate group with them. So this phase is called the phase second, where the splitting of fructose molecule occurs. But important thing here is that in the glycolysis pathway, the glyceraldehyde 3-phosphate takes part in further oxidation. So to maintain this equilibrium, most of time this structure, the 3 carbon structure, get converted into this form. So here you can see that the both have 3 carbon, and this is the aldehyde form. This structure contains the aldehyde function group, and this contains the ketone group. So again the both are isomer to each other. So the enzyme that is responsible for the interconversion of this dihydroxyacetone phosphate to glyceraldehyde 3-phosphate is triose, because both have 3 carbon. Triose, phosphate, both have phosphate, triose phosphate isomerase, as both are isomer to each other. So with this enzyme help, this molecule converted into glyceraldehyde 3-phosphate. So in this phase, the end of second phase, you can say that in glycolysis pathway, the one molecule of glucose at the end of this phase gives 2 molecule of glyceraldehyde 3-phosphate, because this is also converting into this. So one is this and this is also converting. So at the end there are 2 molecule of glyceraldehyde 3-phosphate. Now after that the phase third comes, where this oxidation process start releasing energy in form of ATP. So first I have to write the glyceraldehyde 3-phosphate, and as I mentioned here, there are 2 molecule of glyceraldehyde 3-phosphate, because from this reaction onwards, here every product will be twice. Now further this intermediate is catalyzed by the further enzyme, and it convert it into 1,3-bis phosphoglycerate, and the enzyme that is responsible for this catalyzing reaction is the glyceraldehyde 3-phosphate dehydrogenase. So 1,3, it means here you can see there is already 1 phosphate group on carbon third. So one more phosphate is adding on carbon 1. So there are 2 phosphate on carbon 1 and third, and there is gap in between these 2 carbons. So thereby it is bis. So 1,3-bis phospho and 3 carbon. So that's why it is glycerate. So this reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So where the 1 phosphate comes from? So in this reaction, the enzyme added 1 inorganic phosphate in this reaction, and that phosphate is added on carbon 1. Now in dehydrogenase enzyme, the important thing happens here is the one coenzyme, that is NAD+. It means nicotinamide adenine dinucleotide is reduced to NADH. NAD+ is the coenzyme that is reduced to NADH by this help of enzyme, and this NADH further enters into the ETC, electron transport chain. There this will convert into ATP in form of energy. It will convert, and that can be released through this process. Now after that, the next reaction that occurs and form the metabolite is the 3-phosphoglycerate. So here you can see one is removed, bis is removed. Now the only left is 3-phosphoglycerate. So here you can see that the 1-phosphate group is removing here. So the enzyme that catalyzes this reaction is phosphoglycerate kinase. As I mentioned previously also, the kinase enzymes helps in phosphorylation, that is adding of phosphate group to the high energy yielding molecule. But here the phosphorylation occurs at the substrate level. It means one phosphate group is donating from the substrate and it is adding to the high energy molecule, that is ATP. So here the adenosine diphosphate is converting into ATP. So one phosphate group from this substrate is added to ADP and it is converting into ATP triphosphate, and thereby it forms 3-phosphoglycerate. So again this kinase helps in the substrate level phosphorylation, where from substrate the phosphate group is ripping off and adding to this ADP molecule. Now from 3-phosphoglycerate, it convert into 2-phosphoglycerate. So the name are almost same. From 3 it become 2. It means the phosphate group that is on third will come on 2. So just mutation or rearrangement of phosphate group occurs. So the enzyme that catalyzes this reaction is phosphoglycerate mutase, as it helps in mutation of phosphate group from third to second. After that, the enzyme that catalyzes this reaction is the enolase, because now it is forming the enol. So from 2-phosphoglycerate, here it is forming phosphoenol pyruvate. So formation of enol helped by the enzyme enolase. Now the last reaction. From this, the end product that is being formed here is the pyruvate. So to catalyze this reaction, the enzyme that work upon here is the pyruvate kinase. It means again the substrate level phosphorylation occurs. ADP utilizes this phosphate group and get converted into ATP, and thereby the phosphate group get detached and forms the end product pyruvate. So this is the third phase where the energy get produced. So here you can see from this step every reaction occurs twice. So there are 2 molecules of glyceraldehyde 3-phosphate. So after conversion in this, there are the 2 NADH molecules that get generated. So 2 NAD plus is converting into 2 NADH. Again 2 molecule of ADP converted to ATP. Here also 2 ADP converted to ATP. So in this third phase, every step or reaction occurs twice. So this third phase is called as energy producing phase. And the important thing is here also the 10th reaction, that is from conversion to phosphoenol pyruvate to pyruvate, is the irreversible one. So here this enzyme will not be able to convert the pyruvate back into the phosphoenol pyruvate. So you can see here there are the 3 reactions which are irreversible in nature, that is the first reaction, the third reaction and the 10th reaction. These are the irreversible one. Rest of the steps are reversible one. It means they can change back into the previous metabolite. So in aerobic condition, in the presence of oxygen, the glucose get break down, it get oxidized and forms the 2 molecule of pyruvate here. But in anaerobic condition, when the pyruvate will not be able to enter into the Krebs cycle in the mitochondria, then this pyruvate in cytosol itself get converted into lactate, 2 molecule of lactate. With the help of enzyme lactate dehydrogenase. So again, with this help of enzyme, this coenzyme, that is the 2-NADH, again converted or regenerated the 2-NAD plus. So the energy which is being produced in this step is again utilized here in this step. So here in this glycolysis pathway, we can see that this pathway is regulated by mainly 2 hormones, that is, one is insulin and one is glucagon. So whenever we ingest diet and whenever we take carbohydrate rich diet, the insulin production get increases to break this glucose and utilized by our cell to produce energy. So whenever the insulin secretion increases, that favors the glycolysis pathway, and thus the enzyme that help in catalyzing these reaction, the irreversible reaction also stimulated, that is the hexagon, phosphofructokinase and the pyruvate kinase. So these 3 important irreversible reaction enzymes favored by the insulin secretion, and they increases the oxidation of glucose molecule. But whenever the glucagon production increases, the action of these enzymes get decreases. So glucagon is not favoring the process of glycolysis. It stops the process of glycolysis. Thereby the glucose remain in blood. So this is how our body regulate the glucose production by oxidizing the glucose molecule or disfavoring the glycolysis process. So if we talk about its energetics, how much ATP is generated through this process, then you can see here in phase 1, up till this reaction, once the glucose molecule get oxidized, it uses 2 molecule of ATP. It means it is consuming 2 ATP. But in phase 3, where it is producing energy, here you can see that 2 NADH are producing. These are reducing, and they once get into the ATC, they releases ATP. So NADH is converted into ATP, and it is said that 1 molecule of NADH is giving 2.5 ATP. So as this reaction occurs twice, so 2 NADH is giving 5 ATP. So here 5 ATPs are produced. And again here we can see 2 ATPs are produced here and 2 ATP are produced here. So overall 5, 6, 7 and 8, 9. 9 ATPs are produced and minus 2. It means 2 ATPs are initially consumed by this process. So 9 minus 2 is 7. Thus in aerobic condition 7 ATPs are net produced or generated. But if we say in anaerobic condition where this NADH is again utilized here, then the overall production is only 2 ATP, because 2 and 2, total 4 ATPs are produced and 2 consumed. So 4 minus 2 is 2. So only net 2 ATPs are produced in anaerobic condition, because the whole ATP that is here which is produced, it get utilized here to form the lactate. So in anaerobic, where the end product is lactate, there the net ATP is 2. So this is how we have seen the glycolysis pathway, where the glucose get oxidized and form 2 molecule of pyruvate with the 1 molecule of glucose in aerobic condition and 2 molecule of lactate in anaerobic condition. Thank you.